be presenting our research on the physiology of brain activity during a yawn. Yawning is a behavior that's seen widely throughout the animal kingdom. It's been observed in all 5 classes of vertebrates. Even fish and birds show yawning type behavior. In humans, spontaneous yawning begins as early as 14 weeks in development. On the other hand, contagious yawning doesn't begin until about 4 or 5 years of age. So the prevalence of yawning and also the fact that it shows up so early really suggests that it has a physiological evolutionary advantage as opposed to a social or communicative one. So the brain cooling hypothesis suggests that spontaneous yawning functions as a brain cooling mechanism. So why would this be evolutionary advantageous? Pretty much a cool brain is an efficient brain. Efficient thermal homeostasis has been stressed as an important factor in human evolution. Brains also produce a large amount of metabolic heat and operate most efficiently within a narrow temperature range. Dean Falk has proposed the radiator hypothesis. This hypothesis suggests that throughout human evolution, brains were only able to evolve as large as they could be efficiently kept cool. And here's an example of a radiator-like vascular structure in the brain. And the radiator hypothesis really, Falk is suggesting that as our brains were evolving to be larger and larger, the structure and organization of the blood vessels in the brain had to also be evolving to more efficiently cool our growing brains. So yes, we suggest that yawning is a thermal regulatory behavior. This comes from numerous studies, give evidence to this. Gallup and Gallup showed that yawning decreases with the application of cold packs to the forehead. Also, when their subjects were instructed to breathe nasally, which is known to cool the brain, their yawning frequency decreased. There's another study done in birds. Parakeets were observed to increase their yawning frequency when their ambient temperature was rising. The yawning frequency was also highest during a certain temperature range, so this could be suggesting that yawning is most efficient within a certain thermal window. Another study done in animals was done in rats. Shoutnox et al. Took direct brain temperature measurements from rats, and they observed that temperature of the brain increased before the yawn, and then shortly following the yawn, it decreased. Okay, so here are a couple of graphs from the, it's a follow-up study from the Shoutnox rat study. Here are these are brain temperature measurements. We got temperature measurements from the prelimbic area of the cortex on the top and then the preoptic area of the hypothalamus on the bottom. So in both areas that you can see, before the yawn, there was an increase in temperature, and then following the yawn, there was a decrease in temperature in both areas. The pattern between these two graphs is pretty interesting, and it does suggest something significant. Before the yawn, the temperature in the hypothalamus was increasing sharply. And you can see in the cortex it was more of a gradual increase. But after the yawn, this switches. After the yawn, the cortex experiences a steep drop in temperature, while the hypothalamus experiences a more delayed and gradual decrease. So the preoptic area of the hypothalamus is the main area of the brain that senses heat. It's also the area of the brain that's in charge of thermoregulatory behaviors. So what could be happening here is the hypothalamus is sensing an increase in brain temperature. It's triggering a thermoregulatory behavior, the yawn, and this is cooling the brain, but with a preferential effect on the cortex. So those have been many good studies suggesting that yawning does act as a thermoregulatory mechanism, but there have been no physiological studies in humans investigating this. So we investigate how yawning might function as a brain cooling mechanism. And we investigate the physiological changes associated with spontaneous yawning. And we specifically investigate the mechanisms that may contribute to changes in brain temperature, specifically respiration and circulation. So we extracted yawns from a previous unrelated physiological study from the lab of Ilana Gordis. The previous study was not investigating yawns whatsoever. It was completely unrelated, so we feel that this really gave us a reliable sample of spontaneous yawns. So we confirmed a total of 31 yawns from 27 subjects. We analyzed heart rate, ear pulse, respiratory sinus arrhythmia, respiration period, which is breathing rate, skin conductance, lung volume, and eye closure. And the previous experimenters had already marked yawns in their data set, And this is a common practice in physiological research because physiological researchers know that the yawning can produce some variations in their data, so they use it as a control. That's why they were keeping track of it. So we had to set a definition for our yawn. The first was that it had to be observed and marked by the previous experimenters, but this didn't suffice for itself because they rounded it to the nearest 5 seconds, so it wasn't completely reliable, so we had to set 2 more criteria. And we set these criteria based on the operational definition of a yawn described by Provine. And that definition is that a yawn includes a large inhalation as well as eye closure. So that was our next 2 criteria. Okay, so here is, here is these are 2 graphs, both from the same subject, continuous. Measurements. On the top we have tidal volume, which is lung volume, and on the bottom we have IEMG, and that's measuring eye closure. That's measuring the tension of the muscle and the muscle underneath the eye. So our first criteria was that it had to be observed and marked, and that's these red marks, these red diamond marks right here. That's where the previous experimenters observed a yawn. The second criteria was that it had to have a large inhalation. So this is lung volume up here, and you can see that at both the points where they marked a yawn, there was sufficient inhalation. But the third criteria was eye closure. And looking at the eye closure graph, you can see that there was only sufficient eye closure in this one, and there wasn't any here. So we classified this as a spontaneous yawn and included it in our data set, but we did not include this one. Okay, so using that criteria, we extracted a total of 31 yawns. The time mark 0 is the peak of the yawn, and the peak of the yawn was defined as the peak in tidal volume. And from this exact point, that's where we extracted data for all the measurements. So here is a graph of tidal volume, lung volume. That is, this is the average for all the subjects, all the yawns. So the exact peak in tidal volume was our 0 point. And then we extracted 75 seconds before to 75 seconds after a yawn. So this gives us a 2.5-minute window for data analysis. So one-way repeated measures, ANOVA, were used to analyze each measurement at 5 different time intervals. The first interval was a baseline, and we got the baseline by taking the average of negative 70 seconds to negative 40 seconds. The second time interval was the peak of the yawn, right at the 0 time point. And the next 3 time intervals were 5 seconds after a yawn, 10 seconds after a yawn, and 15 seconds after the yawn. So our first significant result that we think is important in terms of the brain cooling hypothesis is that we saw an increase in heart rate. So heart rate significantly increased from baseline at the peak of the yawn, the 0 time point, and 5 seconds after the yawn. So here's a graph of the average of heart rate for all the subjects. And you can see that this line is where the yawn is. It does increase right at the point of the yawn, and it remains increased for at least 5 seconds. Here's a bar graph of the ANOVA analysis. The asterisk marks significance from baseline. So once again, you can see that it's significant at the peak and 5 seconds after. So we think this is pretty important for the brain cooling hypothesis. As blood is circulating through the brain, the cooler arterial blood is convectively cooling and transferring heat away from the brain tissue it's traveling through. So then that it transfers the heat to the blood, and then the blood flows out of the brain and disperses it through the body. So that's effectively cooling the brain constantly, all the circulation. So if you're increasing your heart rate, then you're going to be increasing the amount of blood flowing through the brain and the rate of circulation. So this is going to cause for more cooling. So we really think that this could be the primary mechanism in which yawning is cooling the brain, coupled with evaporative cooling that takes place in the oral cavity. But this is probably pretty significant for the brain cooling. So our second significant result was that we saw a decrease in breathing rate. Breathing rate significantly decreased from baseline at 10 seconds after the yawn and 15 seconds after the yawn. Here is a graph of the average of breathing rate, our original measurement was seconds after the yawn. The breathing rate was decreased. Here is a bar graph of the ANOVA analysis again, with asterisk marking significance from baseline. And you can see that it was pretty significant at 10 and 15 seconds after the yawn. So this could contribute to brain cooling. Although the breathing rate is decreased, each breath may also be slower and more drawn out. So this could allow for more more evaporative cooling to take place in the oral cavity if there's a longer amount of air passing through the cavity. But more importantly, we think this really refutes respiratory hypotheses for yawning. And respiratory hypotheses are very common. It's, it is a respiratory behavior. The most popular one would be that you yawn to take in a surplus of oxygen, that you're yawning because you need to maintain balance between oxygen and CO2 in your body. So if this were the case, if you yawn and you take in a big inhalation, you're getting a surplus of oxygen, then 10 seconds later your breathing rate is going to decrease, and that surplus of oxygen is going to be negated. If you're not breathing as much, you're not taking in as much oxygen. So we really think that this refutes respiratory hypotheses, and this also goes along good with a study by Provine that showed that yawning frequency did not change in subjects when they were breathing varying levels of oxygen and CO2 mixtures. So that study, along with this, these new results, we feel that it really refutes respiratory hypotheses. So many disorders are associated with impaired thermal regulation. A few examples of such disorders are multiple sclerosis, epilepsy, and migraine headaches. Some of these disorders are also associated with abnormal symptomatic yawning. And Gallup et al. Showed that multiple sclerosis patients experienced an abatement of their symptoms following a yawn. So this stresses the necessity to take into consideration abnormal yawning in clinical diagnoses and clinical settings. So in conclusion, yawning causes several physiological changes that can be conducive to brain cooling. The increased heart rate at the point of the yawn increases blood flow and convective cooling, and may be the primary method of cooling. The decreased breathing rate argues against any respiratory hypotheses, and it could also contribute somewhat to brain cooling. So we're currently doing a replication study. We are another physiological study. It's taking all the same measurements, but we're also having our subjects do a mild mental stimulation in the form of a simple online game. And we are also measuring their temporal artery temperature with an infrared thermometer scanner. And we're currently in the middle of this study, but we have some preliminary evidence showing that that temperature does decrease following a yawn. So I'd like to give a special thanks to Melanie Shout-Knox. I'd also like to thank Gordon Gallup. They are the co-authors on the paper. I would also like to thank the Gallup Lab members, Alana Gordis and her lab for the original data, and Ewan McNay for the animal experiments.